Hi, this is Mr. Carter, your neighborhood-friendly science guy. Today I'm here to talk about the cell cycle. The cell cycle is a complex set of four stages and is extremely regulated by checkpoints and internal and external signals that help tell the cell to stop and go. This provides efficiency and safety for the human body. The cell has four major stages. The first, G1, in the blue near the right, stands for gap 1. This is required for cell growth and getting ready for DNA synthesis. Next is the S phase, which is the maroon red color, and this stands for synthesis, which is where the chromosomes will be replicated. Lastly, the G2 phase, which is in the green on your chart, and this stands for gap 2. This is also needed for cell growth and preparation for mitosis, such as the G1 phase. The last phase is M, which is the purple in the middle and yellow on the outside. And this stands for mitosis, in which the cell will duplicate the chromosomes. There are also many checkpoints along the cell cycle. First is the G1 checkpoint, which evaluates the cell for damaged DNA and other external factors to the cell. If some conditions are, say not met and not able to pass the checkpoint, the cell will end up in the G0 phase, which most of our cells go in anyways, which is on the right of your screen, right of your diagram. This step just means the cell will never replicate. Next is the G2 checkpoint. The G2 checkpoint makes sure the chromosomes have been replicated in the S phase and is not damaged. Lastly is the M checkpoint. This determines if it is ready for mitosis. It determines it by making sure all sister chromatids are correctly attached to the spindle fibers, because once the cell enters into anaphase, it cannot come back out. Next is the diagram, which shows how factors such as PDGF or platelet-derived growth factors trigger signal transduction pathways that allow cells, commonly fibroblasts in this case with PDGF, to get through the G1 checkpoint. It provided the go signal for the cell. Without the PDGF, there will be no division, as you can see right here. But with the PDGF, there will be, as in right here. This helps increase division in the cell, and in a real life scenario, it is also when you get injured and you get a wound or a cut, platelets release PDGF near the cut, which allows proliferation of fibroblasts and helps the wound. This next cycle shows MPF or M phase promoting factor, helping cyclins and CDKs pass through the G2 checkpoint. Cyclins are made in the S phase and start accumulating. Cyclins are a certain type of Cyclins are made in the S phase and start accumulating in that area. Cyclins are a type of protein that get its name from its fluctuating concentrations. These cyclins come together with recycled CDK complexes. CDK are also known as cyclin-dependent kinases because this kinase is only active when connected to cyclins. This produces MPF, which you see right there. When enough MPF is produced, it is able to pass through the G2 checkpoint and begin mitosis. MPF helps start mitosis by phosphorylating various proteins. During anaphase in mitosis, the cyclin is degraded and gets destroyed, ending the M phase. The cell then enters G1 phase and the CDK is recycled for future, and the process starts over. Another terrible thing is cancer cells. They do not follow any sort of regulation. They grow even though the cells are overcrowded. They grow without any type of substratum. They may grow excessively and start invading other tissues. The insane thing, though, is cancer cells could grow infinitely if given a steady supply of nutrients. They are literally immortal. In the picture, you can see in the cancerous tissue the agglomeration of an abnormal amount of cells, and in the regular colon tissue, the proper amount. This mess up in the cell cycle control, if not treated properly, can result in death. Thank you, and I hope that was helpful.